Adarsh Ambati. I am a senior from Archbishop Midi High School, and my project is called Designing Region-Specific Mutation-Resistant RNA Therapeutics for COVID-19 using machine learning and correlative analysis. My project has three major stages. The first stage is running correlations between each of the 12 COVID-19 genes and the mutation rates for these genes against environmental and demographic factors. The second stage is using these environmental and demographic factors to potentially predict for which region across the United States will experience the highest or lowest rate of mutation for each of these 12 COVID-19 genes. And finally, the third stage is using the results from the second stage in order to determine RNA therapeutics that specifically target genes that are less likely to mutate. Just quickly running through the code of my project. Here you can see the processing and the pre-processing stages. Now I'm just going to quickly move into the correlation code. Here you can quickly see the code that I took to correlate each of the environmental factors and the total viral genome mutation rate. And here it is for the E gene as well. I did this for all of the 12 genes. Now quickly moving into the predictive models. Here is the predictive model for the E gene, and here you can clearly see I predicted it with an accuracy of 87%. And here's the ROC curve, had a suitable area under the ROC curve, and the PR curve and had a suitable area under the PR curve. Next, moving into stage 3, as you can see from stage 3, I used the M gene model as a proof of concept, and because the M gene found that Alaska has a low mutation rate for the M gene, I developed 22 siRNA molecules that specifically target the low mutating Alaskan M gene, and so these molecules are less likely to become obsolete, and so they're really good therapeutics. And I believe that through these 3 stages, we can transform the entire field of epidemiology and build a much more stronger and resolute United States against the rapidly mutating COVID-19 genome, as well as any other rapidly mutating pathogen. And I really believe it can transform the entire field as a whole. Thank you.